you want to sit here? That's probably easy. Whatever's easier for you, Mary. You didn't want to lather here?
My thing's too flimsy.
All this works, whatever is comfortable. So welcome everyone to the lunch. There's no break here. Of course, there's lots of excitement, so we cram in as much as we can. So while you're eating away and enjoying your lunch, it's really my privilege today to introduce my colleague, Dr. Merit Sakovits, who for many doesn't need an introduction. She's just been a tremendous face in the world of ALS and has done tremendous work. She is currently the Chief of Neurology at MGH, and I know with a very, very busy schedule, I always worry that she won't have quite as much time, but she's always available. Co-Director of the Neurological Clinical Research Institute, Co-Director of the Muscular Division, and in fact did a BS in Clinical Engineering at MIT, then her medical degree at Harvard, and a master's in clinical epidemiology. I highlight that because really there couldn't be a better qualified person to take on board the activities that she does and the teams that she mentors for clinical trials in ALS. Merit has been a tremendous support for me in helping me develop programs at the ALS Association. She really helped me see the first clinical fellowship program through the American Academy of Neurology. She helped build the concepts of the clinical pilot studies program together with colleagues at NIH. We are excited about the partnership that we have with the Northeast ALS Consortium for Clinical Trials, and we have, through that, funded Treat ALS NEALS Clinical Trials Network. I really think that this has been an incredible opportunity to help build infrastructure and continuity, really, for clinical trials in ALS. We are now also partnering this year and have put out a joint call for a clinical pilot study, and so we're excited to partner on that. So it really is with tremendous enthusiasm and appreciation that I welcome Dr. Merit Sakovits today. She has many of her team with her here. Devon Punch I introduced earlier. She has a tremendous senior team as well who are involved in research, and we fund, and it's going to be tremendous for you to hear from her the landscape of clinical trials. After the presentation, we will have time for Q&A, so we welcome all your questions and thank you for your continued alertness through the program here.
Thank you, Lucy, and thank you, everyone, for listening to me during your lunch. Please continue to eat. I'll eat afterwards. I'm going to plan to talk for maybe 45 minutes and then leave another 45 or so for questions for Lucy and myself. Lucy gets all the hard ones, and I'll take the easy ones. It's really a pleasure to talk today. It really is an era of kind of a revolution in therapeutics in ALS. Taking kind of the look of where the field has gone in the last 10 years, it's skyrocketed. Just purely the number of people studying the disease, the number of ideas, the fact that we're in trials of stem cells, gene therapy, and multiple drugs in this field, and that we know how to do the trials is really huge. This didn't really exist a decade ago. And when companies come to us with their ideas for new therapies, they're just amazed that the whole system is set up for them. So they don't need to start all over again or try to develop the outcome measures or figure out how to even do it. That's all developed, and that's really in large part thanks to the Treat ALS network from ALSA and from a lot of work throughout the US, Canada, and our European colleagues. So I think we're really primed for the next drug, the next stem cell, and the next treatments for patients. So developing treatments is really a collaborative effort. It's between patients, their families, between funding organizations, the scientists, and the clinicians, and the other healthcare providers. So I wanted to take a moment before my talk really just to thank the people that I work with. We have a wonderful group of now 100 centers throughout North America that work really closely together to bring treatments together. That's us on the beach. We don't spend a lot of time on the beach, but we do meet many times to really go over where the landscape is and what therapies we should bring forward and how we can continually improve what we do. The last couple of years, we've had patients and their families at our meeting with us. We are learning from them and vice versa as we really try to break down the barriers and move things forward fast. And we have a team of great scientists that meet now monthly that really review the landscape out there of what treatments are under development, what's ready to bring to patients, so that we really leave no option behind. So I mentioned that we're in a good place. I really do truly believe that. There's a lot of new theories out there on what could go wrong in ALS, and we need those ideas. We actually really need to understand the biology of ALS to really get the treatments. So that's the number one thing that's really critical, and that's what's changed a lot, and again, in the last couple of years. And I got to talk a little bit about that. And we've made progress on therapeutics. We have a long way to go, but we've made progress, and we have 2 drugs on the market, one to slow down the disease, riluzole, one that treats an important symptom called Nuedexta, and we have much improved care. So when people look at how patients have done over the last decade, patients are living longer today, and they're living better quality of life today, and that is in large part because of the better care as well as these treatments. We also have this huge pipeline of therapies, and I'm going to go over many of them. I might not hit all of them because there's a lot of them out there, but if I haven't hit one that you have a question about, please ask me, and if I know anything about it, I'll share that. And as I mentioned, we have an abundance of experience on how to do trials. And I asked the FDA representative about the white paper because I do think that there's enough knowledge out there to be able to really share broadly with everybody what do you need to do to get a drug approved in the United States and Europe. And a lot of that knowledge is out there. And it's important that we share it broadly with people. Nevertheless, we have some challenges, but I think challenges that we can overcome. It's still considered a rare illness, so lobbying, like you guys have been doing for the last couple days, is really key to keep this on the radar for the funding agencies. We're still diagnosing patients too late, and I'll talk a little bit about some of the efforts to try to improve that. We have to get our hands around the heterogeneity between patients, why some patients have the illness 10, 12, 20 years, and why some less than a year. And when we can understand that, I think we're going to make huge inroads into developing new treatments. And just like for any other illness, it still takes a long time to get from your initial idea to on the market. And we have some really innovative ideas of how to shorten that idea, that whole approach that we're working on with the ALS Association, the FDA, and other groups to try to speed it up for ALS. So that's kind of the overview of where I think we are. I'm going to talk a little bit about the changing view of ALS as a syndrome rather than as one disease, and how that impacts therapeutic development. A little bit about the biology and where the targets are, and then really dive into the treatments out there. My slides are going to be available on our Niels ASA website, which is www.alsconsortium.org, and on the ALS website. So if I go fast, no worries, this will all be available. So one of the key changes over the last couple of years is thinking of ALS in the spectrum of a neurodegenerative disease rather than a neuromuscular disease. So neurodegenerative disorder means an illness that as one ages, one part of the brain starts to not function well. And the other neurodegenerative diseases are, for example, Alzheimer's disease or Parkinson's and Huntington's disease. And it's really important to think of these disorders as related and to learn from each other, because the field is really moving that all these diseases are probably abnormalities of protein misfolding or protein aggregates. They might be different proteins in the different diseases in different parts of the brain, but that solving how to prevent the spread in one disease might really end up helping in these other diseases. So a lot of centers and universities are putting all their researchers to study those diseases together, and a lot of meetings now are really cross-fertilizing between these fields so we learn from each other. We've really learned over the last couple of years that ALS is very heterogeneous, and that's really what's making people think it's not one disease. Just like hypertension is not one disease. There's lots of different reasons people have high blood pressure and lots of different treatment approaches. Not everything works in everybody. It's the same thing in ALS. It's probably not one disease, and we'll probably have to start thinking about different treatments for different groups of patients with it. We're learning from the genetics that ALS can be associated with other illnesses, sometimes with a frontal temporal dementia, sometimes with an inclusion body myositis, a muscle disease, sometimes with a bone disease. And learning about this, again, is going to help us get better targeted treatments. The other thing that's important is that even though we consider it a rare disease and it meets all the definitions by the FDA and the funding agencies of a rare disease, it's actually not that uncommon in that if you look at someone's lifetime risk of ALS, it's pretty high. So for males, it's 1 in 300, and for females, 1 in 800. And that just goes along with the fact that it's an illness that gets more common as people age. So there's been a lot of studies to try to understand the risk factors for ALS, and these are really geared to try and understand ways that one could intervene to prevent it from starting in the first place. So we know that genetics is part of it, and you're going to hear all about that tomorrow. But the other risk factors are, as I mentioned, age. So it's more common as you get older. Gender, smoking. Those are the ones that are really kind of secure risk factors that are found in many studies. Some of these other things on the list have been found in some studies, but not in the others, such as soccer and athleticism. There's also some studies looking at what prevents the illness. Though having a high BMI is bad for other medical illnesses, it is a lower risk. People who have a higher BMI have a lower risk of getting ALS. There's also been studies suggesting that people who lifelong take vitamin E or carotenoids are also at lower risks. So these are also important clues and potential ways to intervene in therapies. So in the last 30 years, as I mentioned, there's really been an explosion of scientists studying this illness. And this is just a slide that shows the number of papers per year. So we're up to 1,400 papers a year, and that just shows the sheer volume of science going on. We now know 25 different genes that cause it, and as I mentioned, we have multiple drug trials going on, we have multiple stem cell trials going on, and ways to fix genetic defects. And that's really where our revolution is at the moment. These are all the drugs that have been tried to date in people with ALS in what we call phase 2. So you heard from the FDA representative that there's different stages to drug development. Phase 2 is the hardest part of drug development in neurology. This is where you're trying to figure out what's the best dose, is it safe to give to people, does it do what you think it should be doing in people, and is there any hint of efficacy? And for neurology in general, this is a hard area because we don't have easy tools yet to measure a drug's effect in the nervous system. If you think of, for example, oncology or cardiology, they have easy readouts. So they'll know in their phase 2, their drug either shrinks the tumor or it doesn't. They have something visual. We don't in ALS have something visual like that or something easy in the blood. For another example is cardiology. They can see if their drug in early studies affects cholesterol or not. They have a test they can look at. So there's a big need in ALS to try to develop that kind of biomarker to really rapidly speed up this early drug development. There's a lot of efforts going on, and I think in the ALS newsletter you have out there, there's some biomarker updates from Kevin Boylan and others with neurofilament. But today, those tools to develop those surrogate markers or biomarkers are finally coming in neurology. Better imaging technology, better ways to look at CSF or spinal fluid and blood. And once we have that for ALS, I think it's really, really going to rapidly speed up processes. So on this slide, I put in bold things that have been positive in this phase 2 part in patients with ALS. And there's 6 drugs that have been positive in phase 2. Now, 4 of them have gone on to further testing in people with ALS in phase 3 and have not been positive. So that is a situation one wants to avoid. You don't want to get to the last phase of drug development and have your drug not work. And so there's a lot of things that we can learn from that of how to improve our phase 2 so that we increase the odds and the chance of drugs working when they go to these larger hundreds of patient trials. So the ones that have not gone further and been positive are tolophenol, ceftriaxone, dexpramipexole, and lithium. So that leaves 2 other ones here on the list that have positive phase 2 data that have not yet gone into phase 3 testing, and that's tamoxifen and MP001. Now, the tamoxifen study is going to be reported next week, another study of it. So there was an initial study by Dr. Ben Brooks in about 60 patients, and he saw a very large effect on survival. So about a 2.5-fold in prolongation of life expectancy in patients on the highest dose of tamoxifen compared to people on the lower dose. So that was a very small study, 15 people per group, but nevertheless a finding that people thought needed to be repeated. So instead of leaping to another phase 3 trial, the kind of community decided we should do another phase 2 study and see if that was a real finding. And so in that study, which is done by Dr. Advocacy and other people at Niels, they compared 2 doses of tamoxifen, 40 and 80 milligrams a day, to creatine, and looked at how people did. And the initial readout on that is that people on the tamoxifen dose had a slower rate of decline than people on the creatine. And again, they're going to present all the details of that study next week on Thursday, a webinar through the Treat ALS and Niels program. So I don't have all those details yet, but those results will be available soon. And I'll talk a little bit more about NP001, since I know there's some questions about that in a couple minutes. So I want to get back to what we're trying to do to speed up this process, because I know from the FDA meeting and from talking to my own patients that this is really like critical to take down any barriers that might slow down bringing treatments forward. And so we've done a lot of things with the help of the ALS Association. One is having a network of centers everywhere so that patients can have a center where they can be part of a drug study close to them. So these 100 centers do cover a large territory. It doesn't cover everything, and I did hear in the last session that there are some people who would like to be part of a study but don't live close enough, and we need to get more creative about how to break those barriers down. There are some initiatives to try to do home visits. So one of the recent studies had nurses going to the home for visits. There's also a telehealth using technology and home visits through video conferencing that the ALS Association is supporting us to develop. So I think we're going to keep trying to lower those barriers. But there's basically the trial infrastructure is all there. The sites know how to do it. We're trying to get the access. We're training and teaching patients and families to be research advocates at our Niels meetings. And we're also trying to train lots of other investigators to know how to do trial design. And this is really important. We have held now annually these training sessions, and there are about 30 now investigators out there throughout the U.S. who know how to design trials, and they are now the leaders. And that's really important, again, because we don't want the bottleneck being that we don't have enough ALS clinical investigators. And that used to be the problem, but it's not at all. And the way I kind of measure it is I used to not be able to find any neurology resident to want to come and be an ALS clinical researcher. Now we're getting at least 5 or 6 applications a year. So people have viewed this now as a field that they can make a difference and an impact, and they're coming to it, which will only help us speed up the process. The other thing we're trying to do to lower the barriers is to share resources. So we partnered with Prize for Life to get all the databases of every completed trial in ALS and make that an open shared database. And so that's up and running, and there's been tons of people already accessing it, from companies to academics. And we're also working now, as you heard from Lucy, to really try to push the field forward by making a special call for grants for a company or an investigator who is a drug ready for people with ALS, who can really do a well-designed phase 2 study. And by well-designed, I mean that they know a way to measure that their drug gets into the brain and does the biology it's supposed to do. And we're hoping by kind of putting some resources behind it and insisting that that's the only thing we're going to fund, that we'll get some creative ideas. And so those grants are due in 2 weeks, and I think we're going to get some really good ideas from that as well. The other thing we're trying to do is get rid of the time from when you have an idea and you want to get into people to getting all their approvals at the different hospitals. So this is something called Central IRB. We've already built this for a different initiative for all neurological trials funded by the government. and we want to bring that to ALS. And the idea here is that if you need 50 centers to do a trial, that they all agree that there's one hospital that does the ethics review, meaning making sure that it's safe and ethical to do. You don't need all 50 of them doing it. And this has already cut down time from usually a 9-month startup period to 30 days. So it's really quite phenomenal and something imperative to do for ALS as well. And so we have a proposal, and no pressure here, to the ALS Association to help support that and bring that to ALS. So I want to go back a little bit on why our phase 2 trials challenging in ALS and other neurodegenerative disorders, because ALS is not alone in having these phase 3 failures. Just this week there was another Alzheimer's disease phase 3 failure. It's true in Alzheimer's and stroke. It's more common in neurology than any other field, and it really comes back to this lack of biomarkers or lack of ways to measure short-term efficacy. We want something that you give a drug to a patient in a month, and you know if it affects that target, it's going to prolong their life or slow down the disease. So that's one challenge, and the other is we need to understand this heterogeneity between people, because we really think that patients are going to need different drugs, that not every drug is going to work in every patient the same way. So in the phases of drug development, it can be split into two ways. One is the exploratory phase. That's in the phase 1 and phase 2, when you want to learn everything you can about your treatment in patients, so that by the end of phase 2, you really know the dose, does it have its effect? And you can have multiple of these, and they can be small studies, they can be medium-sized studies, they can be in people in any stage of the illness. And we've heard loud and clear that there are a lot of people who want to be in studies that feel like they can't be in studies. And these are studies where people really could be in the trials in any stage of the illness. Once you have all that data, then you're really primed to do a very good, what's called therapeutic confirmatory phase 3 and 4, where you're really going out to make sure that your drug works and to get it on the market. So some of the key things in this learning phase, this phase 1 and 2, you have to understand the safety of your drug. You might be willing to accept a lot of risks, but you need to learn what those risks are, and that's one of the goals of these early studies. You have to know how to give it and how much to give it. Do you give it by vein, intravenously, orally, in the spinal fluid? You have to know if it interacts with other drugs. If your drug interacts with something else that lowers its concentration, it's not going to end up being efficacious. And also know that it's doing the biology that you think it should be doing. That's called targeted engagement. And again, this is kind of the theme of this recent call for grants, that we want people to really try to do this. So I'm going to give a couple examples of phase 2 studies that have not succeeded, and try to point out why I think they didn't work. So I think everybody was devastated when dexpramipexole didn't work, because the phase 2 study, which was in about 100 patients, explored 3 dosages and showed in the people. We saw a trend, but it didn't reach what's called statistical significance, but it certainly was a trend. And it also looked like it had a trend in survival. So the challenge there is everybody wanted to do a biomarker. We wanted to be able to tell if it was actually working on the mitochondria, or the energy producing parts of the cells, which is what the target was. But there's no marker of that. There's no way to actually measure that in people, in the blood or the spinal fluid or the brain. So even though people knew that that's a good phase 2 thing to do, it wasn't actually feasible. So the decision was made, and it was a risky decision, to go forward to phase 3 just based on having this trend or this hint in patients. So the phase 3 was a large study. Doug Kerr's picture on there, but he wasn't alone in deciding to do this study. There was a whole really community decision to go forward on this, and this shows that there really was no effect on this combined score of survival and function. So we also looked at breathing and strength and all sorts of measures, and we had some predefined subgroup analyses and really did not see anything. So this was disappointing, but in retrospect, we went into this phase 3 without knowing everything about our drug. So we didn't actually know what dose we needed to have a biological effect, because those tools weren't there. to do that. Now, we have still been looking at the data, and I know there's some chat out there about this, so I wanted to bring it up. So one question is, well, there's a lot of questions. Why didn't it work? So probably the number one reason is because the drug doesn't work, and our phase 2 study was too small. It was underpowered to be able to give a definitive read on efficacy. And if you think of it, if we're using the same outcome measures in phase 2 as phase 3, and we need 900 people in phase 3 to tell if the drug works, there's no way we're going to be able to tell in phase 2 with 25 people per group that it works. So we need to do different markers in phase 2, again, these biomarkers, not use the same things we're doing in phase 3. So that's probably the primary reason that the phase 2 was just what we call underpowered, had a very low chance of seeing a real finding. The other things that people have been talking about is, well, maybe there are different patients that enrolled in phase 3 and phase 2. So we did look at that, and they are slightly different, even though the inclusion criteria were the same. So it does look that people in the phase 2 had a faster course. There are people with a more severe course. That's measured by when they came into the study, they had more diffuse disease, what we call definite, meaning involvement of arms, legs, and bulbar. So there were more patients who had kind of widespread illness when they entered that study than in the phase 3. And we're still looking at it, but the challenge is as you start doing many, many comparisons, which has now been done, you increase the chance that you're going to find spurious findings, and it becomes very hard to interpret. The other thing is to try to use the blood samples that a lot of the patients, all the patients donated in the study, to try to see if you can pick out any responders. And those are studies that are being planned, but I think in the big picture, it's very unlikely that we're going to find a subset of people that responded, and that we're better off really devoting energy and resources into doing better phase 2 studies with other drugs that are going to hold more promise. So we ran into almost the identical problem in the ceftriaxone study. So this study was designed a little differently in that it's called an adaptive design, where you do all phases of drug development in one study. So you keep looking at the data in between, and you adjust the dose and other things based on the data, but you don't publicly announce the data. And this way, you can actually speed up the drug development by not stopping between phases. So after the first 60 patients finished 20 weeks, there was a look at the data by an independent safety monitoring board. And in that group, they found that also, just similar to the DEX, about a 38% slowing of the disease, high dose versus placebo, with a dose-dependent effect. And that's here shown on your left. And that was statistically significant. So based on that data and some safety data, the committee picked 4 grams, the higher dose, to go forward in the phase 3 part. But when we finished the phase 3, while there still was a difference that favored ceftriaxone, but it was only 8.2% and it was no longer met the statistical significance. So the p-value, for example, of a p-value of 0.20 means that there's 20% chance that that's not a real finding. And usually the criteria to get a drug approved or for people to believe the drug works, it has to be less than 0.05 or less than 5% chance that it's a spurious finding. So here again, we had a theory of how ceftriaxone worked based on the lab work, that it helped get rid of this glutamate or helped improve clearance of glutamate. But we didn't have a way in people to measure that. We did actually try. We tried to develop a platelet assay. We tried to develop a way to look at it through nasal biopsy, but we didn't have one that worked. So in retrospect, it probably would have been worth doing more phase 2 studies and trying to make sure that we're at the right dose and that you're actually hitting the biology before moving forward to phase 3. So the other phase 2, and this has not gone on to phase 3, is MP001. And I just wanted to bring this out here because I think it's an important decision time for the field of what do you do with this data? Do you go forward to phase 3? Do you abort? Do you do another phase 2? If one had a voice in it. So this is a drug that helps basically against inflammation. So it blocks the activation of these cells in the blood called macrophages. And when this was by Neuraltis, they had 3 groups, 35 people on placebo, 35 on low dose, 35 on high dose. And when they looked just at rate of functional decline, there was no difference between the groups. All right, and that's in terms of ALS functional rating scale as well as breathing. But then they went and looked, well, maybe are there responders? And how they defined responders are people that over a 6-month period don't change at all in their disease course. And when they did that, they found that in the group on placebo, 11% of people were what they called responders. And that number is actually very similar to what we see in all our trial databases. And these are the people that we call the slow progressors who don't change at all in their disease in 6 months or a year. Then they found that 19% of people on the low dose and 27% in the high dose. So this is intriguing. It was not statistically significant. When they just compared it in the trial, it had a p-value much higher than that in the 0.5s. But when they compared it to a large set of natural history control data, they got a p-value of 0.14. So again, a 14% chance that this is not a real finding. What they don't know is, if this is real, what's different between these 27% of people and the other people on that dose that didn't respond? Now, there are blood samples from this study, and in theory, there would be ways to at least try to see if you could get a handle of, is there something different in their biology that explains it? But at the moment, those samples are with the company. Now, I don't know a lot of the details of what the company's planning. I know that they are looking for a partner, because they're a small company, they don't have the funds to go forward. But I would argue that this isn't enough to go to phase 3. The last thing we want to do is another large study, not knowing who the responders are, is this the right dose, does it hit the target? But we might want to do another phase 2, where we really try to identify, are there a group of people who respond to this, who are they, and how can we pick them out? So if I had my choice, that's what I would do. But at the moment, it sits with the company. So what are the challenges? So you're going to probably get the theme that I think one of the biggest challenges is the disease heterogeneity between people. I think we're taking a lot of different diseases into one trial, and that's a big hurdle for a drug to be able to show success. So we have to get smarter at understanding different groups of patients, and I think we're getting there. I think we're getting the diagnosis way too late. Besides the fact that it's horrible to go through months and months of testing and not knowing what you have, it's also probably raising the barrier to getting effective treatments if we're starting later, though we don't have any definitive proof of that. I think our hardest challenge remains the uncertainty of the cause and the targets and how they differ between patients. The brain is a hard organ to get drugs to and to measure effects. It's designed to keep drugs out, but again, the technology is changing, so that's getting easier to do. And then this lack of biomarkers. So I don't want to depress everybody over lunch. I think we can all overcome these, but we have to know what the challenges are to know where to put the resources and the efforts on. So this is just a graph of data from people in the placebo group from one of our trials. And it just shows, again, that on the right here, that about 10 to 15% of people in the course of a 12-month trial don't change at all in their disease, which is great news. But on the other end, we have the same 10 or 15% that they change 3 or 4 times faster than average. And when people come into the study, we actually don't know where they are in the spectrum. We don't have very good prognostic markers. If you think again of oncology, if you have breast cancer, you go in and they say you have node positive, this type of breast cancer, they can tell you your whole prognostic outcome. And they can also tell you which drugs you're going to work better on. That's where our field has to go to. We have to develop those prognostic markers and then figure out which drugs are better for which people. So that's starting. And I mentioned this Prize for Life, Neil's collaboration, where we got access to all these available databases from all past trials. And they put out a competition for people to mine the data and come up with a formula, a prognostic formula for ALS. And they got lots of applicants. They didn't get any neurologists or nurses. They only got computer IT people applying for this. But that's good. You're bringing in new people to the field. And the top 4 winners of the prognostic prize all came up with similar variables. And so what they found that people who have higher levels in their blood at baseline of uric acid, creatine, and their white count, and people who weighed more had a higher body mass index. If you like to look at all those together, that was a positive prognostic indicator. And so I think that's important for us to learn. It turns out that uric acid is something we have in our body that's a natural antioxidant. Turns out that in Parkinson's disease, another neurodegenerative disease, they have the exact same finding, that if you have a higher uric acid level, you have a slower course. So they're in Parkinson's doing a trial of a supplement called inosine that increases your uric acid levels. And so the Michael J. Fox Foundation is funding that in Parkinson's, but you could argue that would be something that we want to be doing in ALS as well. The other thing that's working also on the prognostic factors is trying to see if maybe there's something in people's genes that predicts their course. And so Wim Robrick and Dr. Brown and others last year reported that a particular genetic, what's called polymorphism, so a particular code in the gene for something called EPH4A, if you carried a particular form of it, you were one of the long survivors, the people 10 plus years. Now, the exciting thing about that is that it turns out that that polymorphism, that little change in that gene, caused a decrease in this protein called ephrin. So it's much easier on a drug level to deal with something that you need to decrease than something you have to try to increase. So they're already, based on that genetic finding, there's companies looking at ways, drugs, to try to decrease people's ephrin levels. And we don't know a lot about ephrin, the protein, but it seems to be important in the axon, so the connections between the motor neurons in your spinal cord and the muscle. So this is just, I think, at the cusp of people trying to figure out these prognostic factors. So they're important for patients, they're important for trials, but they also give clues for therapeutic development. The last thing I want to say in this slide is it's because of this heterogeneity that we need placebos. And I'm sure we'll have lots of questions about that. But because we don't know when we see people what their course is, it's impossible to tell if a drug works or not if you don't have a control group. And these small kind of open label studies or small phase 2 studies, I think are actually slowing down the drug development process. And I can say that because I'm responsible for some of the phase 2 studies that were small. Until we get a handle of the heterogeneity, we really need large studies to be able to tell if there's an effect. So just for example, if we wanted to look for a drug that slowed down the disease by 40%, and we didn't have any way to tell people's prognosis or their course, we need 200 people per arm. And so that's a big study. And that's why when you do these 30, 40 patient studies and you try to conclude anything about efficacy, you're likely to be way, way wrong. So again, I don't want to be negative. I just want to get all the facts out there because I think we can get smarter at how we do this by trying to decrease the heterogeneity or understand the heterogeneity. So when we see patients, we know that they're different. There's some people where it starts mainly in the arm and stays mainly in the arm. It's called flail arm. It's not a very great name for it, but that's what it's called. There's people where it's just in the leg. There's people where it starts in the muscles of speech and swallowing, and we know people's courses are different. So the patients are telling us that there's different forms of it, but also the genetics are telling us that there's lots of different forms of it. So one could argue that there's at least 25 different forms of ALS because there's 25 different genes that can cause the familial form of it, and they're all different genes. There's some themes that come out of it, like some of the genes work on how your body makes proteins, other on how they're folded, how the proteins are folded. Other ones are on antioxidant pathways or transport pathways. I know you're going to hear a lot about this tomorrow. But this is really important. It's telling us about the biology and that there's many different ways to get to ALS. We're just starting to try to look, the field is really just trying to understand these different forms. Like, for example, the C9orf72 form that was discovered by Dr. Trainer and others last year looks like some people in those families just have ALS. Some don't have ALS but have a type of dementia. Some have both. And there's companies working on just ways to work on that form of ALS. So this is a slide that I borrowed from Dr. Trainer, which I think is really important because it tells us that we now know the 66% of the families with ALS, we know the gene that they have, and it's really predicted that it won't be long before they know all of them. And that's just because the whole field of genetics has changed so rapidly. And so there's again, companies and investigators are really targeting ways to fix those genetic defects. So I think we're going to see trials just in those forms of the illness. But we're also learning that some of these genes are overlapping in what we call sporadic ALS. These are people who have no known family history. In particular, the C9orf72 gene, which is now accounting for somewhere between 7 and 10% of sporadic patients. So it's an important test to actually do. I know that we've started now testing everybody for it, but if 1 in 10 people without a family history actually have a gene, we need to know about it, especially since there's drugs coming soon that will hopefully target the gene that we know is causing the illness. Now, in those people, they don't think it's a new gene mutation. They still think it runs in the family, but that the parents, for some reason, never expressed it. So there's an ability for people to not get the illness, which is also important to study because it might give clues to how to prevent that form of ALS. I'll skip that. So I hope I made the case that it's not one illness, and that we, in our trials, need to get much smarter about targeting different populations. And I think it's easiest to do in the genetics, but it's not impossible in the sporadic form either, as we develop better ways with imaging and blood and spinal fluid approaches to understand the heterogeneity. The second challenge I mentioned is that it takes too long to get to the diagnosis. This is a study that Dr. Advocacy did where he looked at the last 300 patients that came to see us of what were the steps. And we found that people were seeing 3 to 4 doctors on average before anyone mentioned ALS. And in that seeing 3 or 4 people, they were getting an awful lot of tests, some that were helpful and some that were not particularly helpful. So that was a huge delay. So that's in blue. That's about a 3-month delay of seeing many different doctors, let alone just the kind of anxiety that causes for people. But even before that, there was 4 months where patients weren't even seeing any doctor. And that is an area that I think we can intervene in as well. So we were working, and we're still working on an initiative with Biogen IDEC and a lot of investigators called the Red Flags Initiative. And the goal there is to do just what they're doing in stroke without causing huge alarms in the country. But basically, what are the warning signs of ALS? And we want to teach primary care doctors this. So if you see a patient with X, Y, and Z, you should think maybe ALS and get them to the neurologist quickly. Don't send them to the orthopedic or the ENT or the spine surgeon because a neurologist can probably make that decision very quickly. So that's one first step. Then the other step would be to try to get the word out to patients. So in order to identify the red flag, what those red flags are, Dr. Tassi and others have looked at large claims data sets from health insurance companies, and they have already found a couple of them. For example, if somebody has a weak tongue with fasciculations or has fasciculations in their hand, those are the kind of things that if you see it as a doctor, you should get to the neurologist quickly. And again, it's just theoretical that the drugs will work better early, but we want to at least give that a try. And I also think that there's other drugs that can work in people later in the course, but we need to at least lower the barrier and get patients diagnosed sooner. So I'm only going to show one slide with all the different pathways, but I wanted to put this up there because what we're learning about the illness is that there's many systems involved. And again, it's not likely the same in every person. And a drug that just targets one of these is probably not going to work. So we think that we need either a combination of drugs or drugs that have multiple properties. And there are drugs out there that can target many different targets at once because there's clearly a lot going on once the disease starts. I don't know if the FDA is still here, but I did want to point out that I don't honestly think it's their fault that we don't have another drug. There's always things that they can do better and go faster and give us this white paper and get patients involved. But the number one problem is the complexity of the disease. We need to really get a handle of that, and we need more research. And it's time, and it's already happening, that a lot of this research has to be in people. You know, to be looking with imaging or looking at people's skin cells or their spinal fluid, so we can understand the heterogeneity in patients. So for a long time, people have been working on mice and flies and worms, and those are really critical studies that need to keep going on. They're what gives us all those targets and those pathways. But at the same time, the tools now to look in patients are there. So this is just a picture from skin cells. So now I think people know that you can take someone's skin cell, make it back into the stem cell it came from, and then make it into a motor neuron. And that's a way to study people's individual motor neurons. And that's yielding lots of good new insights into the disease and being used as a way to screen drugs. And we're just now starting to see some of those drugs coming forward for trying in patients. The other way we can really try to study the disease in people is with imaging. And there's a lot of new tools out there that you can use to look at the spinal cord or look at the brain. They're very safe to try to understand the biology and how that differs between people. Again, these really didn't exist a couple years ago, and they're just going into ALS now. So I know a lot of people here want to be part of studies, and there's one way that everybody could be, and is really by being part of these studies in people, what we call biomarker studies. I honestly think we should be collecting clinical data and biological samples from everyone. I don't think it's impossible. It needs, a lot of the systems are there, the networks and the clinics to do this together, and there's a lot of discussions going on to do that because we need to learn from all of you about the illness. So now I'll go to the treatments, and it's really going on two fronts. Symptomatic, and again, huge strides there in just taking care of patients and helping with better quality of life, and then on things to try to slow down the illness. Just on the symptomatic end, we mentioned Nuedexta for this excess emotionality that works. There's studies of exercise going on. There's studies of nutrition. What is the optimal nutrition that are going on? Breathing, there's a diaphragm pacer, a lot of things that are really helping patients today. But I'm going to focus mainly on the kind of drugs to try to slow down the illness. So this is the current list. There's probably more, some that I forgot. I'm going to talk about some of them in detail, most of the first ones in detail. But I just wanted to give you a sense of the scope out there. The ones I don't have slides on, I'll just mention the last three on the first column. So I think it's been on the web that we want to do a study of what we call immunosuppression drugs. These are 4 drugs that are used currently in the neural stem trial to help prevent the body from rejecting the stem cells since they're foreign. We want to see if those drugs alone might have an impact on patients. So there's going to be a small study looking at that. GM604 is a new study by a company called Genervin. This is an example of a drug that hits many targets. So one drug that has lots of different targets. And they're just going to be doing an early phase 1/2 study and are going to be looking at really the biology. Does it affect those targets? Athcar is a drug that will start in June or July. This is ACTH is another name of it. That's also one that attacks many different pathways, neuroinflammation, glutamate excitotoxicity, protein aggregation. So those are both 2 new phase 2 studies that hopefully in the next month or 2 there'll be some webinars either with ALS TDI or with ALSA and NEALS to try to get word of more of those details. So on the left are kind of a lot of drug studies. On the right, I just wanted to show that we really are doing stem cell trials, legitimate stem cell trials in ALS. There's a brainstorm one in NeuralStem, which I'll give you a little more details on. But there's at least 2 or 3 other stem cell trials that are coming up in about a year. One of them is by a company called Q Therapeutics. Another one with Clive Svensson that he might have spoken about previously here. Those are both in animal safety studies right now and about a year away from patients. We're also moving towards studies just in the different forms of the disease. So there's been a gene therapy approach in familial ALS and SOD1 patients, and there's people gearing up to do that in C9orf72 as well. So I'll start with the stem cells. So I only have 2 slides on it, but Dr. Glass and Eve Feldman have been working on with a company called Neural Stem. So these are field-derived neural stem cells that are injected directly in the spinal cord of patients with ALS. They did a first safety study in about 18 people, and now they're moving to a phase 2 study, and that study is going on in Michigan and in Atlanta. The amazing thing there is that they showed that you can safely inject millions of cells into the spinal cord of a patient. That's never been done before, and they're openly sharing that kind of surgical technique with all these other companies that are developing different forms of stem cells. On the right is just a schematic of what Brainstorm is about. It's a completely different idea. So in Neural Stem, they're trying to inject nerve-like cells in the spinal cord and create kind of a healthier environment around the motor neurons. In Brainstorm, they're taking people's own stem cells from their bone marrow. They're called mesenchymal stem cells. They treat them with a proprietary treatment, which I don't know what it is, but it's a treatment to make those cells secrete things called growth factors. Like they're called BDNF or GDNF. They're proteins that we all have when we're in development that help your motor neurons survive. And so they're using your own stem cells as kind of a delivery tool to give these proteins that might be good for motor neuron survival. And they inject those in the spinal fluid or in the muscle. So Dr. Carusis, who's the lead investigator of the Brainstorm trial, presented the results on the first 12 patients at the AAN last March, so about a month ago. And so there's a couple caveats. I wanted to show you the data. So in the study, 6 people got the injections just in the muscle and 6 people just in the spinal fluid. These results are just from the spinal fluid. By report, there was no effect in the people just in the muscle. So a lot of caveats here. So open label study, small number of people. So we know from just the heterogeneity that it's going to be hard to interpret these results. We also know from an unblinded study that there's huge potential for bias, and bias meaning that the people getting the treatment are going to be hopeful that it might work, and our tests are very subjective, and the people evaluating the patients are going to be hopeful that it works. So almost anything you give to people in that setting will show improvement. So it doesn't mean that this doesn't work, but also this is no way says it works. So I know that there's a lot of interest in this, and we're interested in doing a good study to get an answer, but I don't think this gives us an answer, so I wanted to show that. So what they did is they followed people before, so that's in the orange, for 3 months with a functional rating scale, the ALS functional rating scale, and a breathing measure. And then at the arrow, they gave the injections. And here, these are the 6 people in the spinal fluid. And you know, they report a slower slope. So usually you would also see the variance, like some type of graph that would show you how variable people are. This is just the average. So it's hard to tell here if there are some people who responded and some didn't. But this is the data. It was really designed as a phase 1 safety study. So I think there's no doubt it did achieve that goal, that it's safe. They're now moved on to a second study in 12 patients in Israel where they're going up on the dose. So this was a very low concentration of cells. So now every 4 patients are enrolled. They're increasing the concentration of cells given in the spinal fluid and in the muscle. Now there's no phase 2 biomarker for this stem cell or the other stem cell study that I know of. So you could ask, well, can they measure the growth factors to see if those cells are actually secreting the growth factors in concentrations you want? And they really can't, because it's a small amount and it's diluted in 500 milliliters of spinal fluid. So there's no way really to measure that it's having its biological effect. So this next study, the study they're doing in Israel, is really to define the highest, safest dose you can give. Once they've identified that, they're going to come to the FDA and apply for permission to do a U.S. study. They have not applied to the FDA yet. The current deadline or the current timeline that the company is given is the end of the year or so for this trial, but it really depends on the enrollment and the outcome of the results of the current study in Israel. So I mentioned that the exciting thing in ALS is that we're in stem cell treatment. So there's a lot unknown about which type of stem cell we need to give, how many to give. We're at really the frontier of that, but we're in patients. So I think that's a real positive, and that we're only going to get better. I sometimes compare it to there's a book, The Emperor of Maladies, about the first bone marrow transplants and cancer treatments. And they didn't get it right at the beginning, but they kept at it until they got really meaningful treatments. So I think we're kind of not not right at the beginning, but we're a little at the early stages of stem cells in ALS. The other thing we're really at the forefront in is gene modification. So the fact that we can even do this in people is really amazing. And ALS was the first neurological illness that this approach has been used for at all. So this is called antisense oligo. It's when you take a piece of naked DNA, you put it in the spinal fluid, and it goes into your motor neurons. It finds where the SOD1 is, and it blocks your body from making the bad protein that causes the illness. So since it was the first time ever done in any neurological illness, the FDA did ask that we were conservative, so we treated 23 people. We went up on doses. We didn't see any side effects. This was given in the spinal fluid, and now we're really working to go to phase 2 study in people with SOD1 ALS. I put this picture up because this is the very first patient with ALS I ever took care of in 1994. And I made her a promise then that we would not give up on this treatment. And that was just when the gene was found. And it really took, a genetic revolution with the Human Genome Project to get to the point where we actually have the technology to be able to fix genetic defects in people. Because of this study, the company is also working on other genetic diseases in neurology. They've been able to move forward faster in other diseases, like spinal muscular atrophy with the childhood form of motor neuron disease. They're already up to phase 2 there. And again, they're working on C9orf72 and other forms of ALS as well. So I'm just going to fly through a couple of the other studies that are out there. So benefit trial, also known as cytokinetics or tirasemtiv, is really what we call a symptomatic trial. It's a drug that helps your muscles contract more efficiently, and the idea is, can we give people more strength given their function today? And at least in the early studies of this, which were in like 1 dose, 2 weeks, 4 weeks, it did look like there were increases in measures of hand grip and breathing function. And now they're doing a 400-patient study, and it's 20 weeks, and many of you I know are a part of this, and they're anticipating that enrollment will be done, I heard, in June or July, and it's 42 centers in North America and Europe. Gilenya is a study that's about to start in the U.S. So this is work from ALS TDI. This is a drug that's on the market for multiple sclerosis. It's an immune-mediated treatment, so it sequesters these cells called T cells. And again, it works in the mouse model of ALS. We do not know if it works in people with ALS or its safety. So this first study is really to define the best dose and the safety, and that's not an unreasonable thing to do, because in multiple sclerosis, we know early on that those patients had a decline in their breathing that went away, and since breathing isn't a problem in MS, it wasn't a problem in that population, but we don't know that won't be a problem in ALS. So we thought it was really important to figure out the right dose and the safety before launching a larger efficacy trial. Oh, here. So those are the four sites for the Gilenya study. There's also a study of mixilotine. So this is a marketed drug for cardiac arrhythmia. So this is repurposing of an available drug that might be good for ALS. This is being used because it's a sodium channel blocker. So it blocks hyperexcitability of motor neurons. So we know if we look at motor neurons from people with ALS, they're what we call hyperexcited. They're always firing. And so there's a couple different groups looking at drugs that might decrease that hyperexcitability, and mixilotine is one of them. So again, it's on the market. We know a lot about it. It's worked in some of the animal models. But we don't know its safety in people with ALS, and we don't know the right dose to give. So we're, in this study, recruiting about 60 people, testing placebo versus 2 doses, and trying to see its safety. Again, we don't have a great pharmacodynamic marker of this, a way to know that it works doing what it's supposed to do, though there are some experimental things we could try, and there's some discussions about trying to add in a good pharmacodynamic marker. Another study that's ongoing now is Nudexta. So this is the drug I mentioned that helps with the excess emotions that some patients have. It turns out in that study, some patients were reporting that they noticed that their speech and swallowing was getting better. Just what a kind of serendipitous observation in one trial of another symptom. So now there's a study to see if that is a true finding in patients. And this is happening at a couple centers. These are all listed on our website, as well as the ALS Association website. GlaxoSmithKline has a drug that will start in the U.S. in June or July. So this is an antibody against something called NOGO-A. NOGO-A is a protein we all have in our body that inhibits your motor neurons from growing. So this is a drug that gets rid of the inhibitor to help your body's natural way of kind of repairing motor neurons. So they have only done before a single dose study that showed safety. It also showed, in that study, they did muscle biopsies. They were actually able to show that that dose did actually cause some sprouting of the motor neurons. So they did identify a good dose in that study. Then they launched this study, and in the first 50 patients or so, they did in-depth safety studies of chronic dosing, and now they're launching the rest of the study to look at efficacy. This is a study that does require people to live near center because you have to come in every 2 weeks for an intravenous infusion. This is the GM604, a drug that I mentioned by Genervin, and you probably can't see this. I certainly can't see it, but the idea why I wanted to show you this is because this is a drug that in theory works on lots of different pathways that seem important in ALS, and they're just going to do a small study in 12 patients or so and see if the drug actually affects these targets, and they have a couple different biomarkers that they want to test in that study. So I hope I didn't go too long. Okay. Okay, sorry. Well, this is my last two slides. I think we really have gone far. We do have 2 drugs. We have a lot of experience. And even with negative trials, the field's learned, and I think we can go forward much smarter. I think the key point is not one disease. We've got to differentiate the treatments between people. We probably need to differentiate how we treat people in time. That there might be some drugs that work early in the illness, some that work later. And again, the way to do that. The way to do that is to do these studies in patients. So I think that where we're going is really turning this into a survivable illness. And I hope I've left you with that hope that there's so many people studying this and so much new knowledge that we're going to get cocktail treatments. We're going to get drugs that work on specific cohorts of people, drugs that work on many different targets. And while we're doing that, keep improving the symptomatic treatments, which can also make a huge difference in patients. Thank you.
Thanks so much, Dr. Sokovitz. I thought that was a wonderful tour of all the activity and opportunities and what's going on in a very short time frame. So really appreciate that. And then let's fire away with as many questions as we can get in for, I suppose, the next 15 minutes before we have to break out again. Bear with us because it's hard to see you with the lights, but if you can be sure, there's a mic coming towards you.
So when you're trying to differentiate between the different kinds of ALS, are we modifying the questions in the registry to help identify some of those things?
I think to really be able to do this, we need more than the registry. I think the registry is key, but we also need the samples. We need to be able to be looking at DNA, like at genes, as well as blood samples from patients. So it's not going to be enough to take clinical info. We need both. We need great clinical info that we get in the registry and the biological samples.
And then I just want to say, from last year to this year, this is exciting. The difference in the presentation, first of all, and the information, the new things, is thrilling. Thank you.
There's tons. Thank you. There's tons. Yeah, thank you.
Merritt, we're back over here in the corner.
Oh, yes, hi.
Deb Quinn speaking to you.
Oh, hi.
With the antisense for the familiar family side, a lot of us have been into the pre-fall study for the SOD1. We're losing so many of my family, and now that my son has tested positive, what are the chances of taking these that are the pre-falls and allowing them to be a part of the antisense for the blocking and hopeful that they never end up with this disease?
I think the chance is really great. That's the goal. That's the dream. So while we're working on the treating people today with SOD1 ALS with the antisense, the pre-FAL study that you and others are participating in is to try to figure out exactly how to do prevention trials, and that's the goal, to give that treatment also to people before they get ALS symptoms. We have to figure out how soon to do it, and that's the value of the pre-FALs. Do we need to treat people 6 months before we think they're going to get it, a year, 2 years? So we're working on both in parallel.
I think I might add what is remarkable about the antisense, and which really might be helpful in the consideration of treating for longer term, is that it stays around. It's unlike a drug that you have to take once, 3 times a day sometimes. In some cases, they stay around for a month, and you really only have to treat monthly. How this new moiety, this new chemical entity will work, I don't know yet, but the intention is that you don't have to treat very often. So I think it's going to be potentially extremely feasible to do that.
Yeah.
Hi, I am very excited about all of the advances and these drug trials in the pipeline. I've been in as many trials as I can be.
Thank you.
Have you heard of the Deanna protocol? I just met a pals, and she has been using it and has had some real big changes. I haven't gotten to talk to her in detail, but I knew about it after seeing it on TV, and then started researching it and wondering if there is any, thing being looked into to see if this is a possible clinical trial of sorts.
Yeah, so I have looked at the Deanna protocol, and so I think as the ALS Untangled group. So there's nothing, and looking at the list of what's in there, couldn't pick out anything that looked harmful. So just like Dr. Bedlack mentioned, the shared decision, that's what I've been doing with patients. We don't know at all really if it works, but it doesn't look like there's anything harmful in it. There is a group, I believe in Florida, that's trying to do some animal work on it to see if there's any biology behind it, and kind of doing that in parallel. So as of today, I don't know of any clinical trial planned, but I think there's people trying to kind of look at whether there's some science or other rationale as well.
Now, if you have patients who have gone ahead and are doing it,
I have a few,
do you
but
need
it's
to
hard.
know about them, or?
I do, but it's hard in the individual people to know if something's helping. Unless somebody gets better, then obviously you can. But if it maybe it's doing 20 or 30% effect, I don't think we can pick it up. For example, in the phase 2 dexpramipexole study,
Right,
where we know people had about a 30% slowing, the patients didn't notice it. There was no patient telling us, I feel like this is helping me.
Okay.
So I think it's, once people start something, it's very hard to know if it's helping you or hurting you, which would be the flip side, unless it's something dramatic.
Right. Thanks.
Thanks.
Over here. I was diagnosed in September of 12 after about 3.5 years of symptoms. Have you heard anything about a treatment of low-dose nicotine patch to help reverse some of the nerve damage for ALS or PLS patients who have been smokers?
So that's a really good question about whether low-dose nicotine could be helpful for people. I don't know. I hesitate because I think a patient recently brought me something about that, but I haven't yet had the chance to dive into it. So I don't know if you've heard anything about it. So it'll have to be, I'll have to get back to you.
I know in Parkinson's there's been positive data in some sense, but I don't know how clear the data is in ALS. Of course, we know that high doses are not favorable because smoking is very clearly linked, but I'm not sure how strong the data is for low doses.
Yeah. So I'm sorry, I can't answer right now, but we'll have to take a rain check. I'll get your name after.
Thank you. And I was diagnosed with PLS.
Okay. So there are some new initiatives on PLS going on through our organization to try to start the same thing that happened in ALS, start a network of sites doing trials in that form of motor neuron disease as well.
We have a question.
Hi, Mike Gillette, Austin, Texas. The question is more around collaboration, and it certainly seems like you as colleagues are collaborating and the industry is collaborating more. What can we do to speed things up, to collaborate more, share more data throughout this process? And where in the process can we do that to really just speed up the whole development process?
I think I'd say probably in the fundraising, and the reason for that is again, I think there's a wealth of data sitting in all these clinics, all right, and everybody wants to enter their patients in the registries that are out there. But as you heard from Rick Bevak, nobody has any time, because we see a lot of patients and you don't have the resources. But to really support, maybe not all clinics, but a lot of clinics, so that they have a dedicated person, that every patient that's coming in, we're getting their blood, we're getting their clinical phenotype, and that becomes a shared repository, and that would be huge. I don't think it'd be a ton of money, but it needs a kind of focused plan to do that.
Hi, I'm reading this for Chip. He wanted to ask you, you mentioned about getting the drug started on early diagnosis, and he would like you to define early. I was diagnosed in March, but told I am about a year into the disease. Is that considered an early diagnosis in your opinion? If so, what benefit has been proven and with what drug?
So being diagnosed a year after your symptoms is I think, too late. I mean, so that's about what happens on average. Now, I don't have any data that starting a treatment earlier is better than later, and other than what I think is probably common sense, is that if people have more motor neurons in a healthier state, it might be easier for drugs to have a meaningful effect. So I think as we get more drugs, we're going to gather that type of knowledge.
A couple more questions, because I think we're running back to the next series of breakout sessions. So I know Merritt will be around to personally talk with you.
Dr. Sokowa, just one quick question about the C9orf72 testing.
Yeah.
I've read a lot about that one barrier to genetic testing is the cost involved.
Yeah.
Can you just speak briefly to that in terms of how patients might be able to access testing and have it paid for by insurance?
That's probably a good place to advocate too. So it does cost a lot. So the just getting the C9orf72 is about $300. Getting the whole ALS panel is like $1,500 to $1,800. I think that probably have to advocate that it makes a difference and to get insurance to companies. So some insurances cover it and some don't. Sometimes we've used our clinic money for that to help people. So I don't have a great solution, but I do think that it's much more common than a lot of the other tests we do. You know, we send Lyme, we send hair samples for metals, immune studies. None of those has a 10% chance of being positive. So I think it's almost part of the diagnosis, but we probably have to write papers on that and get something in the literature peer-reviewed so we can convince insurance companies.
I would just advise in the interim, I can't see the person who asked the question anymore, sorry, but in the interim, until that's resolved, I would encourage you to get involved in research studies, because then the cost is paid, and we support and fund research studies in this area amongst multiple investigators, so we would be happy to see if we can do that for you. One more question, I think. Okay. Last one.
Yes. My name is Dan Chapman. I'm from Denver. And I asked one of the previous speakers about this. He was making a point about the lower registration numbers for clinical trials.
and I suggested a very low-tech solution to that problem, namely having the sponsoring institution answer their phones more frequently. When you see a clinical trial posted on the internet, you get very excited, and there's a name and a phone number and a email address, and you can chase those around till you're blue, and you will not get a response. In all my inquiries about Project Brainstorm, including your own hospital, the only response was from the Israeli biotech company in Tel Aviv. So I guess my question is, we're knocking on the door, but how do we get in?
Well, there's no excuse for not answering people. I would say that maybe we have to tweet or do something else. No one answers phones anymore. But for Brainstorm, I have to just tell you that we probably get 30 or so inquiries a day, and we don't have the resources to answer all of them. So we've been trying to put things on the website to answer that kind of frequent questions, so that we can try to get back to people as best as possible.
But certainly I would suggest if there is a trial that's up on our website or through the NEALS network is on the site, and they are enrolling patients and that number's not working, I would suggest you email Devon, because she certainly would look into that and make sure if it isn't a working number that site is contacted. So, obviously for more general questions where we don't necessarily have access to the investigators or the PIs, that might be tricky. But for those that we list that are really enrolling, if there's a problem with a phone number, let us know, because that is probably an easy fix. It might even be an error transcribed somewhere, or if not, they must take that off and provide another number. Yeah. So I think with that, actually, we will conclude the session. A really big thank you for Dr. Merkimeritz-Sakowitz's commitment and hard work and presentation. And thank you to all of you for being so attentive. It's been a long day, and I know you'll need to stretch your legs now. So thank you so much, and look forward to presenting more in the years to come on the progress.
I'm sorry, we're over again.